Welcome to another episode of Chromatography Experts, the podcast in which we discuss all things separation science. My name is Anson Lewis, Global Sales Manager at Tosoh Bioscience, and with me today are my colleagues Yuka Kervinen and Fu Duong.
Hello, I'm Fu Duong, Senior Manager, Global Commercial Strategy.
Hi, my name is Yuka Kervinen. I'm a global scientific expert, and my specialty is chromatography at Tosoh Bioscience.
All right, so this episode, we want to focus on one specific topic. It's about cell and gene therapy, Yuka, okay? And I know that you've been focused so much on this topic, right? Not only, you presenting, material, scientific materials, but also, you actively involved, in many experimental designs at Tosoh Bioscience, okay? And I know for a fact, recent years, cell and gene therapy is such a hot topic, okay? So can you explain, what is adeno-associated viruses and why it's so important in cell and gene therapy?
Oh, surely. Thank you for... Yeah, the adeno-associated viruses, or simply AAVs as we call them, they are modified viral vectors filled with genetic material and therapeutic material, what we can use for many inherited diseases to treat them.
I see.
Yeah.
Yes, and I know that answer is, have a few questions for you as well, Anson.
I do. So I kind of want to clarify something that we talked about a bit earlier. Can you kind of go into a bit more? Why are AAVs so important in the space of cell and gene therapy precisely, especially for patient outcomes? Like, what makes this so important?
Oh, yes, as Yuka Chip explained previously, right? Because AAVs, one thing about AAVs, yes, it can pack a lot of information, genetic information, or drug information in AAVs. And AAVs not coming in one specific kind, but many stereotypes of AAVs. As Yuka pointed out, that nowadays that we identify up to, about 12 or 13 different AAVs, okay? And not only that, AAVs, the way is they deliver in the body also is very, very effective as well. Okay, for that reason, it is quite important to use AAVs, because I know that besides AAVs particles, we have other particles such as lentiviruses and many things similarly like that. But those are important, but significant bigger, and more study need to be done. Whereas AAVs has been done for many, many years and more understood.
Understood.
Yeah, and for myself, I think, as I've said, the simplicity is the key. Mm-hmm. They are simple viral vectors, and they certainly have become immensely important in gene and cell therapy applications to treat many inherited diseases and even some acquired diseases. They are suitable for that.
Yeah.
So obviously, cell and gene therapy is a booming space right now, Yuka. So how do you define AAVs in the context of other cell and gene therapy type of vectors?
So in simple ways, there are four main types of gene vectors or viral vectors. So that we have adenoviruses, retroviruses, and our adeno-associated viruses and lentiviruses. And about adeno-associated viruses, AAVs, I could say they have a typically low toxicity, and they have a very good tissue specificity. They find the way very well where to go. And also they have a very good ability to precisely cut the DNA and actually put the new DNA in place. So they work very well. And they are simple viruses.
I see. And the other thing that Yuka, I want to understand a little bit more. I know for the fact that when come to analyzing these particles, AAV particles, presenting a lot of challenges to scientists, right? But the reason why, I think that because of how complex these molecules is, okay? But you recently published a method to analyze, these AAVs effectively. And I know that I read a little bit about this publication. Can you go through a little bit about what you done for AAV analysis?
So we took a project in the laboratory recently where we started studying the AAVs, and we started with the analytical part of the AAVs. So first it's important to understand that the AAV virus is only as good as its therapeutic material, genetic material inside the virus. If there's no genetic material inside that capsid, actually there's no potency. So, and we know that the method how we purify produce the AAVs, the efficiency how you pack the DNA material, it's only 20 to 40% efficiency. So, and you have to know when the virus has the genetic material and when it doesn't have it. So we developed a new method, actually, analytical method, where we can actually study this so-called empty-full ratio of the adeno-associated virus. And we finished this method about a year and a half ago, published a paper about it. And in this method, we hope that it will find its way to the many quality control laboratories and also for the virus development work.
So now, one thing that I want to understand about the new method that you established or the method that you published, right?
Yeah.
What's so different between this method and previous methods that people use now? That why it makes so effective?
Yeah, I would say there are two components. One is our Tosoh column, TSKgel Q-STAT column, and the novel part is using a totally new type of gradient salt. We call it the choline chloride.
I see.
Chlorine chloride is very good salt to test because it has high solubility, it's totally non-toxic, and it gives a very good separation efficiency. Previously, people have been using a lot of another chemical called tetramethyl ammonium chloride, or TMAC. And this TMAC has a very good separation efficiency, but it's hugely toxic to humans. Or even the 3 grams, if you get it to your mouth, it may kill you.
Oh, wow.
So this is now the new method, which gives a new very good separation efficiency, and also improved laboratory safety.
Oh, I see.
So, Yuka, about the column that you use, what makes the Q-STAT anion-exchange column so exceptional to work with when it comes to AAVs?
So I would say Fu could tell also even more than me about this column. You are an expert about column structure, but for me, I would say basically that the column has a relatively good particle size. It's about 7 micron particle size, and it has a quaternary ammonium type ligand there. Together, this column with the choline chloride salt actually works really well. It has a bonding chemistry and also low operational pressure, separation efficiency with this column. It's very good, and that's we selected this column. We tested other columns also, but we certainly found that the TSKgel Q-STAT column works best with this method.
Yeah, I think that if we dive deeply into the column technology, I think that the TSKgel Q-STAT is one of my favorite columns, right?
Absolutely.
It is as you just pointed out, it is truly an anion columns. But one thing about this column that I like a lot, I mean, is non-porous technology.
Yeah.
So people did not have to worry about pore size selection at all, because everything happening, on the surface of the particles, right? So the mass transfer is just very effective, rather than have the molecules have to move across the particles here that are just on the surface. So it moves from one particle to the next a lot more effective. Another thing that I like, that the particle size is not so big, but they're not small neither.
Yeah.
For that reason, you can use your HPLC or USPLC effectively. So I think that, and only that, with the non-porous technology and the way that, we pack these columns, the speed is just incredible, put it that way as well. Yes.
Yeah, absolutely. And if I can add it into that, yes, one assay takes about 25 minutes, or even 20 minutes. You can run the one sample. So it's a speed and a low operational pressure, as I said. They all make that column very, very good for AAV work.
And I was told that in the method that you develop, I mean, not only you can identify the empty versus full, but the resolution was phenomenal.
Yep.
And I remember that I read the article is also you can do some sort of racial differences as well.
Yeah,
Right.
Yeah, absolutely. You can use actually the standard simple gradient to go up, and you can get almost the baseline separation of the empty and full capsids. But you can actually make isogradient steps actually to the method and improve that method, and you can see a total separation of empties and fulls. And you can even see if there's some partially packed capsids, you can start seeing those two with the steps.
So I think that we want to make sure that we can put the link of the paper over the podcast. So if anyone want to read it, they can do.
Absolutely.
So moving on, Yuka, based on your knowledge of the just the general cell and gene therapy industry, what are some key bottlenecks or challenges to AAV production and analytics today?
As I already mentioned, and we discussed a little bit, yes, it's AAV production in upstream is very inefficient process. So as I mentioned, about 20 to 40% of AAV's content is packed genetic material. And apart from the analytical part, even more important is the preparative part. You have to find an efficient purification process in downstream to get rid of those empty capsids and enrich the full capsids. And I think that this is the most important part of the AAV whole work, is to find the way to get rid of empty capsids because they don't have any potence.
Okay. So Yuka, when come to seeing gene and cell therapy, right? Not only, AAVs are important, but I knew that other, vehicles, for example, lentiviruses and things like that are so important. But what makes AAV is so attractive to scientists out there?
I think compared to other viral vectors, they have also pros and cons they can use for different purposes. But I think the main thing with AAV virus, it's simple structure, its workability, because there's only that shell of the particle, and those are the two very good important things to work: workability and simple structure.
Oh, I see. Yeah, but it looks like it's easy to work compared to other. And I mean, they're not easy to work. It is a bit more understood, and then more varieties to work with, right?
Yeah, and I would like to also add one more thing, that actually, compared to some other viral vectors, human body has actually relatively low reaction against the particles. So the immunological reaction, there is immunological reaction like anything what you put to the human body, but AAVs have that actually relatively low immunological reaction in humans. So that's the other thing.
I'm sorry, how about deliveries in the body compared to other particles?
I would say that all viral vectors have two good tropism.
Okay.
It means that they find the way. Some viral vectors find very well, some a little bit less well. There are at least 13 different AAV serotypes at the moment. And those different serotypes are built so that actually they have a different tropism. Some find the liver, some find the brain better, and some find the other organs. So AAVs are very good in that sense, that you can use the serotype which best fits your purpose, where is the genetic defect in your body, and you start using that serotype.
I see.
So looking ahead, where do you see AAVs kind of headed when it comes to clinical development?
I would see that I see the future is bright. So we have, if you look at the literature and other, you follow also news even on TV, you can see that there are more than 200 preclinical and clinical studies ongoing for AAVs. And there are also currently seven FDA or EMA approved therapies available at the moment. So I can see all these together. I see the future very bright for AAVs.
Now, Yuka, I actually want you to disclose a little bit. Okay, I know that you get involved heavily in, AAVs based, work right now. Can you share with people what currently you do in the lab, or you have a few projects in the lab?
Okay, now now it's getting interesting actually, because I'm now really revealing something which is not yet even published actually. So, but I will tell you, moving from the analytical work, we have been recently concentrating on heavy